depending on if you've been exercising, for example, if pH is going to be lower, what have you. And this will interrupt any of these weak interactions, specifically, for example, hydrogen bonds, right? So we can't rely on weak interactions to hold these two together, even though we need them to be together in this inhospitable environment. So what we find disulfide bridges very often is in polypeptides that are secreted, right, to hold them together or to lock them in the correct configuration. Again, because there's changes in pH and changes in concentrations of all different metabolites in your blood, in your lymph system, depending on different environmental conditions. Again, if you've been exercising, if you've been eating, if you've been drinking a lot. So here what we see is that we have one intramolecular disulfide bridge between these two cysteines on the A chain, but we have two interchain disulfide bridges between the A chain and the B chain. And because they're covalent, they're much more difficult to break, right? So we'll always find the A chain and the B chain together, ensuring that insulin remains functional even after it's secreted by the pancreas. Now, peptide bonds are kind of strange, right? We always draw them as this single bond here between the carbonyl and the amino, but really they exist in resonance, right? So similar to the nitrogenous bases of DNA and RNA that we saw on Tuesday, we can draw the peptide bond in either one of these configurations, and both are equally correct, right? So that means that it's not fully a single bond, but it's not fully a double bond either. It's a partial double bond, right? You might remember this from organic chemistry, I'm sure. Everybody remembers everything from organic chemistry. So, what does this mean for the peptide bond? It means that it remains planar. Okay, the peptide bond cannot rotate to any appreciable degree because of that partial double bond characteristic. Remember, single bonds are free to rotate however they like, as long as you don't bump into anything else. Double bonds can't, right? Because what we have is really a superposition between these two, right? This also can't really rotate, okay? So the peptide bond is planar. But the other bonds within an amino acid are not planar, okay? So here, for example, we have the peptide bond between the carbonyl and the amino group, right? So this remains planar. On this corner of, say, some kind of playing card, we have the alpha carbon from amino acid 1, and over here we have from amino acid 2, right? So this kind of remains as this rectangle, kind of a playing card, which isn't fully, twistable. But this bond here, right? So here we have this residue here, alpha amino, alpha carbon, or alpha carboxyl. This is one residue here. This bond between nitrogen and alpha carbon can rotate just fine. The bond between alpha carbon and carbonyl carbon can also rotate just fine. And we can quantify the degree of rotation between these two using dihedral angles, right? So we call the rotation between the N and the C alpha the phi angle, and between the C alpha and the carbonyl carbon the psi angle, right? So phi and psi. For any given amino acid in a protein, it will have its own phi and psi angle, okay? So let's quantify this here. So if we want to look at the phi angle, right? So we'll look at, here's a little I, we're looking at the nitrogen in front from this amino acid, from this residue, to this alpha carbon. That's what we see here, right? We have the nitrogen in front, the alpha carbon behind. And what we're measuring is the angle between the carbonyl carbon that's attached to this nitrogen, not shown here, right? So this is what's over here, not shown, and this carbonyl carbon here, right? That's attached to the alpha carbon behind. That angle is our phi angle. In this case, it's negative 80 because it's going counterclockwise. Similarly, we want to look at the psi angle. Well, now we look at the alpha carbon in front of the carbonyl carbon. What we're measuring is the angle between this nitrogen and this nitrogen, right? So you can imagine that as we're seeing here. We have the alpha carbon in front, the carbonyl carbon behind. Here we have the nitrogen over here. Here we have this nitrogen connected to the alpha carbon. Here we have, for example, positive 85 degrees because it's going to the right. Now, I'm not going to ask you to bring a protractor to the exam, but it is important to understand the dihedral angles because they give us an insight into secondary structural elements. So here what we're looking at is called a Ramachandran plot, and this is for a generic amino acid. All right, so it's just an R group. We don't care what it is, right? So say alanine, which is simplest chiral amino acid. And what we see that there are only certain combinations for phi and psi that are allowable, right? And that's what's shown in the green. Everything outside the green, okay, is not allowed. And why isn't it allowed? Well, if we look over here, okay, in this region that is allowable, right? So between, for example, minus 170 and minus 60 phi angle, and between 70 and 50 psi angle. These are allowable. And why are they allowable? Well, because all of the groups, all of the bulky groups, the functional groups, the amino groups, the carbonyls, the oxygen, they're all far apart from each other. Okay, so they're not bumping into each other, they're not causing any ruckus. Right? And the same over here, this big allowable region here. Again, nothing's bumping into each other, everybody's happy. But once we start to deviate from what's allowed, okay, well now we see that we start to actually cause steric hindrance or steric exclusion. Right, so here we see a steric hindrance. We have this bulky functional group next to this oxygen. They're not going to want to be close together, right? So they're pushed apart until some combination of allowable phi and psi are achieved. Okay. Now, additionally, looking at the phi and psi angles for a given amino acid in a protein, right, gives us an idea of what secondary structural element that amino acid is a part of. Right. Let's see some examples of that just right now. So moving on to secondary structural elements. So what have we got so far, right? We've got a linear chain of amino acids, right? Starting at N and going to C. But as we saw in the beginning, right, we can have amino acids that fold into different shapes, right? We have ones that can look like a beta clamp, we have ones that look like ferrotaxin, right? So, how does that happen, right? And how do we define that, right? How do we get from this linear chain into a defined structure of a protein? So the first step there is the secondary structure. The first secondary structural element that we'll talk about is the alpha helix, right? There are several different types of secondary structural elements. The alpha helix, the beta strand, the beta sheet, the beta turn, and the beta loop. Okay? Let's start with the alpha helix. The alpha helix is always a right-handed helix, okay? Never a left-handed helix. Here's a little diagram for you, right? So how do we know if a helix is right-handed or left-handed? You simply move your hand in the direction of your thumb, right? Here's my right hand, so I'll move in the direction of my thumb. Can I wrap my fingers around the helix as I move in the direction of my thumb? And the answer is yes. You can't do the same for your left hand, right? It won't wrap around, okay? Now what happens if I turn this upside down? Is that a right-handed helix or a left-handed helix? Huh? Right? Yeah, it's still right. Doesn't matter which way you look at a helix, it's always going to be left or right-handed. The only way to change it from left to right is to take a mirror image, which you can't really do, right? Now, if you can't wrap your mind around the right hand and left hand, you can also pretend that this is a spiral staircase, right? And you put yourself at the top of the staircase. If you walk down the staircase and you're turning to the right, then it's a right-handed helix. If you're walking to the left as you descend, then it's a left-handed helix. Right? So here we have what a helix generally looks like. An important thing to consider about an alpha helix is that the functional groups of all of the amino acids that make it up are always sticking out away from the axis of the helix, right? Which means that we can control the properties of a helix by controlling what amino acids are found on the exterior, because they're always on the exterior, which functional groups are found on the exterior. So for example, we might be able to have an amino acid where all of the residues on this side, right, are hydrophobic, and all of the residues on this side are polar. Right, so we can have now what we call an amphipathic helix, where vertically split down the middle, we have half of it which is polar and half which is non-polar. We might find this at the interface between the cytoplasm and the phospholipid bilayer, for example, right? Or inside of a porin protein. Now, an alpha helix is stabilized by weak interactions. All structural elements, except for in a protein, except for the disulfide bridges, are weak interactions. In this case, we have hydrogen bonds holding together this alpha helical structure. Okay, notice the hydrogen bonds are between the backbone carbonyls and the backbone amino groups here, right? So we get hydrogen bonds. Notice that we have our 180-degree bond angle here, 180-ish degree bond angle here, 180-degree bond angle. So these are strong hydrogen bonds between a residue and a residue 4 away. So for example, this carbonyl is forming a hydrogen bond with this amino group 1, 2, 3, 4 residues away from it. Okay? And this over here, right, this oxygen forming a hydrogen bond with the amino group 1, 2, 3, 4 residues away, and so on and so on. This, right, can form a hydrogen bond as well, right? Where would this form a hydrogen bond? At the very end, right here, 1, 2, 3, 4, right here. So we get many hydrogen bonds stabilizing this. And notice that we have this periodicity, this repeating structure to the helix, right? Such that the rise, over here, the rise is always 1.5 angstroms per residue, meaning that there's a vertical distance between adjacent residues of 1.5 angstroms. There are 3.6 on average residues per turn, right? You can't have 0.6 of a residue, obviously, it's just an average, right? So an average of 3.6 residues per turn, right? Notice 1, 2, 3, 4, right? 3.6 would be somewhere in the middle, which is right where we started, right? So it's on average 3.6, right? So that's just an average. So don't get too bogged down with the 0.6 of the residue. But of course that means if our pitch is 3.6 residues per turn and our rise is 1.5 angstroms per residue, that means that we have a pitch of 5.4 angstroms per turn. So it's 3.6 residues per turn, which comes out to 5.4 angstroms per turn. Now, this ball and stick model of an alpha helix is kind of misleading, right? Really what we have is something like this. If we look at the space filling model, again, remember the radius of each of the atoms is determined by its van der Waals contact distance. But what we see is that the interior of an alpha helix is essentially filled, right? So instead of being this empty kind of porous thing that we see over here, an alpha helix, though it is helical, acts more like a rod, right? The interior of the rod is completely filled up, so nothing can really fit in there. But again, we see that the functional groups, all these greens, are on the exterior, facing away from the axis of the double helix. So again, we can control what the double helix is going to do, right, by cutting it down the center. So we might be able to put all non-polar here, all polar here, right? So we might find this at the interface of, I mentioned the phospholipid bilayer before, but we also find amphipathic helices at the extremities of a protein, right? So on the outside of a protein, right on its surface, we often find amphipathic helices with the polar side, with the non-polar side rather facing in, and the polar side facing out for reasons that we don't care. Now, because an alpha helix has to have a repeating defined structure, there are only a narrow band of allowable phi and psi angles that a residue can adopt and still be part of an alpha helix. And we see that here. If we have, a stretch of residues that all have phi and psi angles in this range, then there's a good chance that that is part of an alpha helix, okay? Now the reason for this is because all of the residues in an alpha helix have to bend in a certain way to achieve both the right-handedness and the pitch of an alpha helix in order to satisfy the formation of these hydrogen bonds, right? So the bond angles are very strict. So that's why we see this very narrow band of allowable dihedral angles. Now here, it's kind of interesting, right? Here we have a left-handed helix, which is essentially just the negative of both of these, right? Of the phi and the psi angles. And this makes sense because what is the difference between a right-handed helix and a left-handed helix? Well, they're just mirror images of each other. So we take the opposite side of the dihedral angles, and that will give us a left-handed helix. Now this doesn't really exist in any way that matters to us, all right? This is very rare. It can be made synthetically, okay? But essentially we don't really observe right-handed helices, though it is theoretically possible. And they have been made in the lab and observed in very rare circumstances. All right, now alpha helices can vary, or a protein rather can vary in the amount of alpha helix it can have. It can have no alpha helix, or it can be entirely alpha helix, right? We'll see an example of that in just a minute. 25% of all proteins are just alpha helices connected by turns and loops, which we'll talk about in a minute. Most helices are about 30 amino acids long, which if you remember, 1.5 angstroms per residue. That means that it's about 45 angstroms long, okay, the average alpha helix. Some side chains are not, or some rather, residues are not allowed in an alpha helix for a variety of reasons. The most obvious, right, or at least the most obvious to me, is proline. Okay, why wouldn't we ever find a proline in an alpha helix? We never find a proline in the middle of an alpha helix. Now, what's different about proline? Right, the R group loops back around and connects to the alpha amino group, meaning that the phi angle of that residue is constrained, right? If that phi angle is constrained, then it cannot adopt any bond angle in here. So it cannot ever be found in an alpha helix. Where do we find prolines with reference to alpha helices?
Beta pleated sheets.
Huh?
Beta pleated sheets.
No, with respect to alpha helices. Huh? Yeah, where it bends or at the ends, right? Prolines are helix breakers. We very often find prolines at the end of a helix. Why? Is because once we have a proline there, well, now that's game over, right, for that particular helix. We can form a helix right after it, in which case you're right, we do get a kink, right? Or we can form a loop or a turn and then start a new alpha helix, right? But a proline is very useful as a helix breaker. We can also not use valine, threonine, or isoleucine, right? Why is this? These clash with the backbone, right? These are too bulky, right? They're branched, and so they actually start to make contact with the protein backbone, which remember is what's stabilizing the alpha helix by forming these hydrogen bonds. We also get no serine, aspartate, or asparagine. Reason being that they have high hydrogen bond capability. So they will steal the hydrogen bonds away from the backbone carbonyls and amino groups so that they can't stabilize the alpha helix, right? So we don't find bulky groups. We don't find very polar groups that can form hydrogen bonds, and we don't find proline. These are not found in alpha helices. What we do find are things like alanine, right? We can find, let's see, glutamate, right? Interestingly, glutamate can be found, but not aspartate, because glutamate has a longer side group, right? So it puts that carboxyl further away, so it can't form a hydrogen bond, right? But we can find other amino acids in these alpha helices. Now, moving on, the next secondary structural element is the beta strand, right? So what we see here is one beta strand. Notice here we have from N to C, and we have an alternating pattern of where the functional groups are. So in any given beta strand, okay, which is essentially just an elongated arrangement of amino acids, we have function groups sticking up and then down and then up and then down and then up and then down. Each residue or each functional group is 3.5 angstroms away laterally from the one next to it, which means that two adjacent ones on the same face are 3.5 angstroms plus 3.5 angstroms, which is 7 angstroms. So on the same face of a beta strand, we get an amino acid pointing in the same direction every 7 amino acids, and the same on the top. And similarly to an alpha helix, where we could split it down the middle and say this side is going to be polar and this side is non-polar, we can do the same thing to a beta strand, right? We can say, all right, all of these, every other amino acid is going to be polar, and every other amino acid on the other side is going to be non-polar. So now one face of our beta strand is polar and one side is non-polar. We again find these in porin proteins that we find in the phospholipid bilayer that create a water-filled chamber on the inside to allow polar molecules to slip through the phospholipid bilayer, right? But this makes beta strands very useful. But we don't really ever find a beta strand on its own. We usually find a beta strand in the context of a beta sheet, which is essentially just many beta strands hydrogen bonded together. Again, a beta sheet stabilized by hydrogen bonding, just like an alpha helix is stabilized by hydrogen bonding. And there are 3 different arrangements of beta strands that can make up 3 different types of beta sheets. Here what we see is an anti-parallel arrangement. We have N to C in this direction, but on the opposing strand we have N to C in this direction. And in this case the hydrogen bonding is very simple, right? The amino group from one amino acid directly interacts with the carbonyl of another amino acid, and vice versa. So we get 2 hydrogen bonds between 2 whole amino acids. In a parallel arrangement, notice we're going N to C and then N to C, so they're both going in the same direction. We still get each amino acid, right? So here we have 1 residue forming 2 hydrogen bonds, but not now with 1 whole amino acid on the other side. Instead, it's split. This is hydrogen bonding with 1 residue, and this is hydrogen bonding with the next residue, right? But we still get 2 hydrogen bonds per residue. So they're both equally allowable, right? Not one is more allowed than the other. So here we're just looking at an example of having 2 beta strands. Well, we can have as many beta strands as we need, right? Here's one that has 3 beta strands, right? We can have 4, we can have 10, we can have 15 beta strands. Doesn't matter. They all make up a beta sheet. How many just depends on what that protein is trying to accomplish. Now here what we're looking at is the last arrangement of beta strands, which is neither parallel or anti-parallel. Instead we call it a mixed beta sheet, right? So we have the top 2, which are arranged parallel, and the bottom 2, which are arranged anti-parallel, so it's mixed. Most beta sheets are mixed, right? It's very rare that we find a beta sheet that's purely anti-parallel or purely parallel, okay? Now, how are all of these connected, okay? So we might have, over here, an alpha helix, over here, a beta strand, over here, a beta strand, and here I'm showing you, right? Okay, well, different beta strands can come together and form a beta sheet, but here we just have a linear polymer, right? So how do we get interaction here? So for those, this is how we depict a beta strand, right? Just with arrows in the direction that they're pointing. All right, here again, this is where we find beta strands, again, because they're more flexible in terms of the allowable phi and psi angles, just because they don't have a defined periodicity or defined structure, right? We can have beta strands that are a little bit twisted, a little bit turny, right? So it's a little bit more nebulous than strictly an alpha helix, which has to look exactly the same way each time. A beta strand has more configurations, like you'll see right here, right? We have this, for example, this saddle, or no, this flat beta sheet. We have flat beta sheet, but notice each of the beta strands aren't perfectly straight, right? This one actually makes almost a 90-degree turn. This one's kind of slanted. This one's kind of slanted and twisted. This one's twisted about 80 degrees, right? So these different beta strands, okay, can adopt different configurations, which is why more allowable, or because more phi and psi angle combinations are allowable. Now we can also have differences in how the beta sheet itself is assembled, right? A beta sheet is not completely flat, right, with, nice, evenly spaced, flat beta strands. Instead, we can have things look like this, right? So here's a 90-degree turn version of what we see in panel A. Here what we have is a saddle, right? So we can have twisted beta sheets, we can have barrel beta sheets, right? We can have a sandwich beta sheet, which is what we see there, right? Now, how do all of these secondary structural elements interact with each other? How do all these beta strands come together close so that they can form these hydrogen bonds to make a beta sheet, however it is supposed to look, a saddle, a barrel, a sandwich, whatever, okay? Well, this is accomplished by loops and turns. Here what we see is a beta turn. Beta turn is made up of exactly 4 residues, right? And they're stabilized again by hydrogen bonding between the first residue and the residue 4 away, right? By the backbone carbonyl and the backbone amyl, right? So a beta turn is called that because it makes pretty much a 180-degree turn. So in between 2 beta sheets, right, we might have a beta turn, okay? That would twist this like that. Now this beta sheet will be next to this beta sheet, they can form an anti-parallel beta sheet, okay? We can also have beta loops, right? Beta loops are another way that we can add dimensionality to our protein. Now, beta loop, though it's depicted like this, kind of this, wiggly kind of strands of amino acids out here connecting beta strands in this case, okay? But these are rigid, right? These aren't just out flopping around. These have a defined structure. Now they're not as defined as, say, an alpha helix, okay? But this isn't just all, floppy in the wind, right? They're held together in that structure, in that configuration by weak interactions, like hydrogen bonding, like van der Waals interactions, and so on, all right? So these have a defined structure. In fact, antibodies, right? Antibodies, they have a defined structure at their ends, and that structure is these beta loops. It's the structure of these beta loops that allow an antibody to recognize an antigen. That's only possible if those beta loops have a defined structure so they can recognize the antigen, right? So these beta loops, though they look flopping around, right, they have a defined structure. So all these together, right, allow the secondary structural elements, the beta loops and the beta terms, allow the secondary structural elements to come together and form the tertiary structure, which is how these secondary structural elements interact. So here we have, for example, myoglobin. Myoglobin is made up of 7 alpha helices linked together by several beta terns. Here we see a beta tern, here we see a beta loop, right? But it allows these alpha helices to interact together and form the tertiary structure of myoglobin, right? The tertiary structure, again, describing how the secondary structural elements come together to form the final structure of a protein. Now, what drives this process is interesting, right? If we had just a polypeptide chain, right? We had just a polypeptide chain and we had to go through every possible structural iteration of how those say 200 amino acids can fit together, well, we'd be here a long time, right? It would take it would take that polypeptide billions, if not trillions of years to go through every possible iteration until we found the correct one. So what drives the formation spontaneously of protein structure? The main force doing that is hydrophobicity, right? So here again we're looking at myoglobin, but in this case the polar amino acids have been colored blue, and the non-polar amino acids have been colored yellow. If we take a cross-section like this, right, so we cut it in half that way and look at the interior of myoglobin, we see that there's a high concentration of non-polar amino acids on the inside, and a higher concentration of polar amino acids on the outside, right? This is what effect is this? It's the hydrophobic effect, right? And why does that matter? Well, this is all happening in the cell, right? So say here we have the cell. The cell is made of mostly water, right? So all those polar amino acids, right? They don't want to be in contact with the water, like we talked about on Tuesday. So where's the only place that they can go? Well, onto the inside of the protein. So now we have our globular protein. Here we have the non-polar amino acids on the inside, right, being pushed together away from the water. And on the outside, we have all our polar amino acids. This allows the protein to remain soluble in the aqueous solution that is the cell, right? And that's what drives the formation of the tertiary structure. It's what defines how these secondary structural elements are going to fit together, right? To get all those non-polar amino acids on the inside. That's further stabilized now, right, by weak interactions between the secondary structural elements themselves, right? Hydrogen bonding, van der Waals interactions is a special type of the hydrophobic effect anyway, right? So after, right, we get kind of driven to the rough conformation by the hydrophobic effect, and that's stabilized by the weak interactions. All right. All right. In the interest of time, I'm going to skip through. Keratin and collagen. All right. So we have those on your slides. Just skip through those. I'm going to skip through keratin and collagen. I'm going to skip I'll hit domains. Right, so we'll get to domains here. I do want to talk about domains. Now, once we have the tertiary structure of our protein, okay, the tertiary structure of our protein can have different what we call domains, right? A domain is a region of a folded protein that has a defined structure and a defined function. So we might have an amino acid, or might have a polypeptide, let's say, right, that folds into a protein, but part of that protein, part of that sequence, is what we call, for example, a kinase domain, meaning that that domain in that polypeptide has a defined structure and has a defined function. In this case, a kinase, something that transfers a phosphoryl group from ATP onto something else, right? You don't have to know that right now, right? But a kinase has a defined function, and there can be a domain within a polypeptide that has a defined function. There might be another domain on that same protein that, say, recognizes a substrate, right? So now we have a substrate binding domain and a kinase domain on our polypeptide, right? So this, we would say, all right, well, this binds this substrate through this domain, and then it does something with the ATP to phosphorylate it maybe through the other domain. So different domains can work together. And how do we determine if there's a domain? Well, the sequences of many proteins has already been solved, and the function of those same proteins has already been established. So we look at those proteins, we see what they have in common, and say, oh, well, these all have this particular amino acid sequence in common, and they're all kinases. So maybe this is a kinase domain, or specific kind of kinase domain. So if we see that in another protein, maybe that's also a kinase domain. So this is all kind of prediction, right? And going through is what we call bioinformatics, right? Looking at data and trying to predict future outcomes, right? And what we see is that, yes, that is true. If we have similar sequences of amino acids that we see in other proteins that do similar things, then that's likely a domain that does a specific job in that protein, which helps us predict what that protein does. I'm not going to go over motifs either. Okay, so domains are important to know. Motifs, we're going to skip over just in the interest of time. Also, collagen and keratin, we're going to skip over because I do want to get to the quaternary structure of proteins. The quaternary structure of protein is just one last level of organization. Now, not all proteins have a quaternary structure, okay? Some proteins, after they've formed their tertiary structure, they're done and they're fully functional, like, for example, myoglobin, right? Myoglobin only has one polypeptide chain. That's that example that we were looking at just before. And once those alpha helices come together and interact, then myoglobin is good to go. A similar example that does a similar job but in a different way is hemoglobin, right? Hemoglobin does have a quaternary structure. And what is the quaternary structure? It's just different polypeptide chains that have already adopted their tertiary structure coming together and interacting to form a fully functional unit. So not all proteins have a quaternary structure, right? All proteins have primary, secondary, and tertiary. Not all of them have a quaternary structure. But if a protein does have a quaternary structure, it must adopt the quaternary structure. For example, hemoglobin is made up of 4 individual polypeptide chains that have already folded. We have 2 alpha chains and 2 beta chains that have already folded to adopt these conformations. Either the alpha chain on its own or the beta chain on its own is unable to bind oxygen. It's only when 2 alpha chains interact with 2 beta chains, making a heterotetramer. When they interact and form hemoglobin, now it's fully functional, right? So an enzyme or protein that has a quaternary structure must adopt its quaternary structure in order to function. All right, so how do proteins actually adopt this structure? Okay, well, it was observed that proteins adopt structure. They fold by themselves. They fold spontaneously, right? Nothing else is needed, right? In most cases. Let's just stick with the general case. Nothing else is needed in order for a polypeptide chain to fold into its correct structure. So what does that tell you about the thermodynamics of protein folding? Which is more stable then? The polypeptide chain or the folded polypeptide? Why?
Because it's spontaneous. It's going to have no purpose.
What's going to happen with those?
So the folding is going on.
The folding is spontaneous, right? Which means going from unfolded to folded is spontaneous. So which one is more stable, the folded or the unfolded state?
Folded.
The folded state, right? If it's spontaneous, that means that it's going towards its lowest energy state. That process is going towards its lowest energy state, meaning that by definition, the folded state must be in its lowest energy. So how does this happen? How was this discovered? Well, this was one of the seminal experiments of biochemistry called the Anfinsen experiment. I'm going to go over it quickly, and I'm going to cover every single detail, but just to illustrate the point here. So here what we're seeing is the amino acid sequence and the rough structure of an enzyme called ribonuclease. Notice that it's got 4 disulfide bonds holding it together, okay? And this is, again, the rough structure of ribonuclease. And what ribonuclease does is very simple. It just degrades RNA. And this was useful because the amount of RNA in solution is very simple to measure, okay? So if we add ribonuclease to a solution of RNA, Anfinsen was able to measure in real time that the amount of RNA was decreasing, right? Which makes sense because that's what ribonuclease does. Now, what he did next was to destroy the tertiary and secondary structure of ribonuclease, but leave the primary structure in place. And he was able to do this by adding 2 things. He was able to add beta-mercaptoethanol and urea. Beta-mercaptoethanol is a reducing agent, right? Why do we need to add a reducing agent? Well, because, remember, we have 4 disulfide bonds in ribonuclease. And that's holding it covalently in the correct configuration, right? So it's going to stabilize the structure. So we need to get rid of those. Remember, disulfide bridge is only formed between oxidized neighboring cysteines. So by reducing those cysteines, we break those disulfide bonds, right? So beta-mercaptoethanol does this. The next thing that he added was a denaturant, in this case, urea, right? So urea, we're going to see again in the urea cycle. But urea, what it does is essentially negate the hydrophobic effect that drives protein folding. And how does it do this? Well, if we look at the structure of urea, we have 2 hydrogen bond donors here, 2 hydrogen bond donors here, and 2 hydrogen bond acceptors here in this oxygen, right? Now, what does this do, right? Well, this essentially hydrogen bonds with water, right? Sequestering it. So effectively, what urea does is, in effect, lower the concentration of water. And if you remember, what drives the hydrophobic effect? Well, it's water, wanting to hydrogen bond with itself. But now they're hydrogen bonding with urea, that impetus is no longer there. So by adding both beta-mercaptoethanol, the reducing agent, we break the disulfide bonds, right? So we loosen up the structure. And then adding urea negates the hydrophobic effect that caused it to adopt the correct configuration in the first place. So what happens now after he added denatured and reduced ribonuclease? Denatured is what we call a protein that we've destroyed the native configuration for. So denatured reduced ribonuclease. Well, now we have this random coil, and when we add this to a solution of RNA, what Anfinsen discovered was that RNA is now no longer degraded. So this established that, yes, it is the structure of a protein that gives it its function, right? The primary structure is still there, right? All these are still held together by peptide bonds. We haven't done anything to the peptide bonds. We're no longer any structure, no longer any activity. Now the question was, can we rescue this ribonuclease? And the answer is yes, right? So if we add, or if we rather, remove remove the urea, followed by the beta-mercaptoethanol, right? Well, now we've removed the urea through dialysis, right? Don't get too bogged down with dialysis is. You might already know from chemistry, but either way, right? We remove the urea via dialysis. This allows the hydrophobic effect to return, causing it to adopt more or less the right configuration. And then again through dialysis, we remove the beta-mercaptoethanol, which now allows those cysteines to reoxidize and form those disulfide bridges. Okay? So now the question was, all right, let's remove the urea and the beta-mercaptoethanol. Can we take that now, add it to the RNA, and will it degrade RNA? The answer was yes, right? So after it was renatured, added to a solution of RNA, he was able again to observe that RNA was degraded. The amount of RNA went down over time. And he didn't do anything else except for remove the reducing agent and the denaturant, right? So it was able to fold spontaneously on its own. And so what does this tell us about the structure and thermodynamics of protein folding? Well, it tells us that all of the information necessary for the 3-dimensional structure of a protein is already encoded in the primary structure. The sequence of amino acids themselves is sufficient to drive the correct folding of the protein. Nothing else is needed outside of some exceptions, right? But the sequence itself encodes structural information, all right? Now, how does this process actually look like in real life? Right? Well, we don't have this kind of linear relationship between the folded and the unfolded state. And we were able to discover this by using different types of spectroscopy, not important right now, but suffice to say that there are methods that we can use to determine if a protein is folded or unfolded without doing all these assays of, degrading RNA and what have you, right? So here we start with a fully folded protein, okay? And what we do is we add denaturing in increasing amounts, right? So here we have the concentration of denaturing. And what kind of some people were expecting were, all right, the more denaturing we have, the more unfolded protein we'll get, right? Kind of in this linear relationship. But what was observed was that we essentially don't unfold anything until we reach some threshold, when then all of a sudden we get mostly unfolded protein, right? And if you look right in the middle of that threshold, right, which they did, and they examined what the structures of those proteins are. In other words, how folded are those proteins? Well, they didn't find, a mixture of fully folded, 25% folded, 50% folded, 75% folded, and 0% folded. What they found was that at the midpoint here, they still had proteins that were either completely folded or just completely unfolded, which means that folding and unfolding must happen all at once, right? And that's what we see here and here, all right? Here's a simulation, a computer simulation of probably how proteins fold. Now this happens on a time scale that we can't really observe, right? So the best that we have is to look right now at computer simulations. And here's a very short polypeptide. Now here we start in the denatured state, and we want to end in the native state. What happens is that first we get the formation of some discrete secondary structural elements, for example, an alpha helix, right? And as soon as this alpha helix forms, this acts as a nucleation site, meaning that when this alpha helix forms, it makes it easier for nearby regions of the polypeptide to also adopt their secondary structure, right? So here we get more secondary structure being formed, which acts as more nucleation sites for more secondary structure, which then adds for more and then adds for more. So as soon as we get the first hint of structure, this quickly collapses down to its lowest energy state, which is the fully folded protein, which is what's shown here, this kind of energy funnel. Okay, here where we have a high degree of energy, right? So we have unstable, just a high degree of entropy as well, right? We get the beginning of helix formation, for example, right? So we get the formation of the first alpha helix, and that drops the energy down, right? And acts as nucleation sites again for other secondary structural elements, which are now in their more stable configuration, so the energy drops down again. Those serve as nucleation sites, so we get a drop down in energy again and again and again until we fall and collapse all the way down to our native structure, right? This happens again on the order of probably nanoseconds. It's very, almost basically impossible to see in real life, but this is basically what's happening, right? As best as we can tell, right? All right, so there is protein structure. I did want to get to misfolded proteins and prions, but I guess we'll have to skip that.